It's so great to be with you today, and appreciate everyone who's, I guess you didn't come out to join us, but you're wherever you are in your homes joining us. I know these are difficult, stressful, challenging times, so I appreciate that you're spending some of that time with me and with the Science Museum. And a big shout out to the Science Museum during these times. We certainly are looking for scientific information to inform us about these challenging circumstances. So it's really great to be a part of this lunch series. So as Jim said, I've been at this in this discipline, a university and college professor for a while, looking at different topics, mostly focusing on rats. Today I want the talk is a little bit different, a little bit lighthearted, so that we can have some fun. So trying to do several different things. Since we have a theme about driving and trips, it's always good to have a plan to map out our journey. So just to let you know what I plan to do during our short time together is to talk a bit about brains, because they're one of my favorite things to talk about, because they're so awesome. And then how experiences leave an imprint on brains. And then we will get to probably what you're looking more forward to hearing, how we can kind of combine some of those interests to look at teaching our research program where we're teaching rats to drive little cars, rodent-operated vehicles, ROVs. And I have learned a lot, not only about driving, but about the brain with that research. Along the way, we will have a rest stop after before we start the rat races. And I think Jim's going to be collecting, if you have questions through the chat, we'll take a few minutes there to answer the questions about the brains and experiences before we go to the rat races. So that's our plan. So I work because I'm very interested in brains, and it's easier to look at rodent brains than human brains. So we use models for brains, and the rat brain is one model that I have used for most of my career. And you see one of these rats here. I have the utmost respect for these animals. They're smarter than me most of the time, outsmart me. You see that the rat brain here, it's quite small compared to the human brain, about 2 grams. Our brains are about 3.5 pounds. I wonder about mine quite often, but it's supposed to be that big. So a rat brain is not a human brain, but it is a good initial model to study some of the aspects of human brains. They share many characteristics. There are a few that are different that we're becoming more sensitive to. But we do a lot of that work so that we can hopefully translate it to the human brain. And you see this very complex, awesome human brain here. We scientists, neuroscientists, act as detectives to try to figure out what the parts of the brain do. What is the function of this area of the brain, the cerebellum hanging off the back of the brain that's involved in motor coordination versus all these squiggles, this cortex up here. There's so many mysteries, so you can certainly be a lifelong detective when you're interested in the brain. But we hope that some of our work with models can translate to human conditions, looking to see, what are the underlying mechanisms of stress and depression and anxiety and schizophrenia. We have a long way to go. I'm the first to admit we haven't made a lot of progress when it comes to effective therapies, especially cures for these psychiatric illnesses. So we want to know more about healthy, problem-solving, adaptive brains. A little bit more about the bits and pieces of brains. I'd like to give a shout out to my colleague, Dr. Susanna Herculano Huzel. She did most of her work and training in Brazil, and she recently, a couple years ago, she's now at Vanderbilt University. And she's very resourceful, and she came up with a technique called isotropic fractionation, in which she literally blends, homogenizes parts of brains to count how many cells are in those brains. So if you're a student of the brain, or you've been going to brain kind of movies, or movies where they talk about, futuristic movies about the brain, for a long time we read that there were 100 billion cells in our brains. You couldn't count that many, but it sounded good. It was an informed guesstimation. Well, her work with this blender, a blender-like and brain soup, as she calls it, has confirmed that the human brain has 86 billion neurons instead of 100 billion. And a difference of 14 billion, that's an entire baboon brain. So that's a large margin of error. So she has done so much to tell us about the cellular composition of brains, not only human brains, but across the board, so many different animal brains. She's also recently written a book about the human advantage and what makes human brains different. And again, they're on a continuum with all the other mammalian brains and primate brains, but they are somewhat special in the number of cells. Humans don't have the largest brains, and they don't have the brains with the most neurons. But in that outer area, the cortex, just beneath our scalps here, we do have exceptionally dense populations of neurons. And that makes us different in some way. We're special on the continuum. So she has made the point, I recommend her TED Talks. If you Google brain soup and her name, she'll talk about that technique. She also has another TED Talk where she discusses the importance of what we do with all those cells. It's not good enough just to have 86 billion neurons. We need to shape those neurons into circuits. And that happens through education and through our experiences. So this pile of Lego blocks is pretty useless, but if we build them and form them into meaningful structures and circuits, then they're quite powerful. So it's our experiences that lead us to be able to build these circuits. And here's just an example of how beautiful the brain is, and how all these cells, and I've just told you the number of neurons, but there are other cells like glial cells in the brain, and they're all forming connections. And when you're a student of the brain, you wonder how any of us end up where we're even semi-functional with all of these circuits that need to be created for us to be functioning individuals. I am a neuroscientist, but my home is in the psychology department here at the University of Richmond, and it's always been in the psychology department. I was at Randolph-Macon College for almost 28 years before coming to the University of Richmond, so I've had the distinct honor and privilege of being at two wonderful institutions within the state of Virginia. But I've been reading recently about a psychologist, James Gibson, who wrote about this term affordances, and how they offer the animal a different perspective of something in our environment. For example, if based on our environmental culture and experiences, when we look at this little teapot or tea kettle and cups, we know immediately in a handle, we know immediately what we can do with that object. We can pick this up on the handle. We know something's going to pour out. We know that we can pour it in this cup and pick this cup up and sip that. But we're not born knowing that. It's through our experiences. So with rich experiences, we can look at something out in our environment and consider these affordances and know what we can do with it, or to experiment with what we can do with that. So that enriches our lives. But if we are not exposed to an enriched environment or a complex environment, we're going to be limited about what we see that we can take advantage of with objects in our environment. So a child, based on his or their experiences, will look at that pile of Lego blocks and through different perspectives of affordances see, I can build an animal or a ship or a car in alignment with today's topic. Those experiences are driving the brain to interact with that environment in that way. So experience is so important. And in my lab here at the University of Richmond, we're interested in what I call experience-based neuroplasticity. So that is what imprint or fingerprint or footprint does our experience leave on our brain. So kind of have a fingerprint overlaid on this brain. But the brain has all these squiggles. I mentioned that in the cortex that makes the human brain somewhat unique. Other animals have a cortex, but we have a lot packed into that cortex. And it's those squiggles, we call them gyri, or the gyrification, that allows us to pack a lot of brain real estate into our smallish area of this head size that we have. So if we had a big piece of aluminum foil, we can compact that and squunch it up and get it into a smaller area. So that is what nature, that's the pattern, the prototype, the design that nature has used. And we all have very individualized gyrification in our brains. So our brain surgeons need to keep us awake, or scalps are anesthetized, so the surgeon can see, okay, what is this area of the brain doing on you? So that if we need to damage that, we know what we're dealing with. So the individual differences are just amazing. So how do our experiences map on to the neuroanatomy, the neurophysiology, and even the neurochemistry of our brain? It's just a fascinating story. I know several weeks ago, if you've been coming to the lunch series, Dr. Catherine Franson, who Jim mentioned, talked about the importance of enriched environments and natural environments. And I was very fortunate, speaking of experiences, for Dr. Franson to have worked in my lab as an undergraduate. I've known her for a few years, and then as a postdoctoral fellow. So maybe that work had some imprint on her brain, and her experiences with me had an imprint on mine. But we have enjoyed looking at different types of enriched environments. So much of biomedical research is done with what we call a standard environment. Rats are placed into a very basic cage. They have, it's not shown here, but they have all the food and water they want, temperature regulation. So it's a nice, safe environment. It's kind of like a Hotel 6. It's not a resort. What we're interested in is to put them in more complex environments. And we've learned that comparing a more artificial, where there are manufactured objects in the environment, to more naturalistic environments, they have some qualitative differences. So we kind of call this the country rats and the city rats. They both cognitively seem to be pretty similar, but when we look at stress and emotional resilience, something we all need now, we find that the country rats, these more naturally enriched animals, seem to be more resourceful and more resilient. So when we look at stress hormones and such, they have a healthier profile than the rats in the more artificial enriched environments or the standard housed rats. So we've done a lot of work with those environments. One concern I mentioned that most research uses that standard environment. And I want to say, no, we need to all be using enriched environments, because that probably gives us a more representative view of a brain, the rat brain. Rats, they're living in this challenging environment, maybe in our houses, maybe out in our yards, cities, wherever. They're very adaptive. So we need to represent that more in the lab if we want to have results that can indeed translate to humans. And that's what the National Institute of Health is interested in, mental health. So we've tried to solve this, and my students and I, to make an enriched environment that may be a little bit more practical. Those enriched environments take up a lot of space and they're messy. So we call them the rat lofts. They only take up twice the amount of space, and we can take them apart and wash them. And it gives them the experience of travel. My students and I, we tried to kind of pare down what is incredibly important about an enriched environment or about an animal's natural environment. The ability to travel is one of those aspects. So even though they have a limited environment here, to be able to travel from one cage to the other, and it's like a little subway. Speaking of individual differences, the first day these were installed in these animals, you see that there are 2 or 3 in each cage. One filled it up. It could be a hole that a snake would come out of, so they're being defensive. One's not so sure, fills up half of it, and the other's like, I'm going down. So they're almost genetic clones. They've had very similar experiences and environments, Even so, when faced with this new challenge, each cage acted very differently, but they can zoom up and down. It's exercise, it's exploring. So we're having a good time looking at the differences in those environments, comparing it to an enriched environment to see if we would get similar effects, and also that standard environment. This is one challenge. We put them in something called an open field and then put a predator odor, a cat odor here in the middle. We don't actually put the cat. And then we had a hiding place. And I thought, using my human brain that supposedly has all these neurons in the cortex, that the smartest response would be to hide in the hiding place if you thought a predator was there to threaten your life. Well, the animals in the lofts and the enriched environments, they were more likely to jump on top. And I thought, duh, if you went inside the hiding place and there really was a cat in there, you would be stuck, right? Busted, you're going to be eaten by the cat. But by jumping on top, it enhanced their vantage point, their view, where they could actually see if there's a predator in there. So I thought that was brilliant. We also was fascinated by the fact that none of the standard rats did this. So in the standard cage, they hadn't had experiences where they could travel or jump up and down like the other enriched animals and the loft animals did. So it wasn't even in their behavioral repertoire to jump up there. So the affordances, perceived affordances, were very different with this stimulus if a rat was raised in a standard environment, a loft environment, or the enriched environment. So if our world is restricted, like it has been in quarantine and isolation recently, it may start to influence how we see the world when we go back out to it. But hopefully we don't have cats threatening us and life is good and things are going just as expected, and it's interesting to look at the brain during those situations. But life is life and things happen, and a lot of times we need to recalculate what is going on. And I think this is the best way to look at what a brain can do. So just quickly, before we get to our rest stop, we do have some protocols where we train animals and influence their experiences where they can build connections between their rewards and their effort. So we're using kind of education, behavioral cognitive therapy. If I dig in a little pile, I get a Fruit Loop, and they love Fruit Loops. Other animals are just given the Fruit Loop. So we've changed the experience where this contingent trained animal, the Fruit Loop is contingent upon the Fruit Loop, and it eats it makes those connections versus the non-contingent animal. So the animals that dig up their Fruit Loops, those are our worker rats. Students like to call the others the trust fund rats. And we like to see how these experiences change their affordances or perspectives of the outside world and stimuli. So when we give them a task where they no longer need to dig up a Fruit Loop, but we put one in a little ball here, they've never experienced this. The worker rats in purple here, they'll persist longer, trying to bite at it, throwing against the wall. The trust fund control rats, they're perfectly able, to interact with the stimulus, but they give up sooner. And also with our contingent trained animals, they have lower stress hormone, healthier stress hormone profiles, so that we think that we've made connections in what we're trying to do among the areas of the brain that are involved in reward, like this area called the nucleus accumbens, if you're a brain nerd, areas of the middle part of the brain involved in movement, and those in the cortex involved in decision making, so that we can build our own neural circuits like those Legos, so that when we do experience some uncertainty, that we're able to have some sense of agency and control and solve those problems. An area of the brain called the hippocampus is one of my favorites. Looks like a beautiful garden, doesn't it? But our brain's ability to change, also referred to as neuroplasticity, we look to the hippocampus to see evidence of how the brain is changing, how these Lego blocks are being redistributed and forming new circuits in this area of the brain. And we do see some neuroplasticity changes with our contingent trained animals, and also with enriched animals. And they have, as I mentioned, the lower, healthier stress hormones. Stress it's a predictor of a lot of, psychiatric illness. So it's important to reduce stress when we can. I hate to say that during a pandemic. So Also, behavior-based or behaviors can change our neurochemistry. A target of a lot of the antidepressants is serotonin that's involved in our emotions, emotional processing. It's also released during repetitive behaviors. I have knitting here, so that could influence serotonin. Dopamine, as we knit something that we value, that's known as a feel-good neurochemical. That's oversimplifying, but it also alerts us to things in our environment that are important. And oxytocin, if we're doing this knitting with a group of people or friends or maybe even a cat sitting next to us, that cuddle chemical is increased. So we can change not only our neuroanatomy, but neurochemistry through our experiences. I like to call this behaviorceuticals instead of pharmaceuticals. So hopefully I've convinced you that our behavior and our experiences can change our brain with our work with the rat. I would like to say that we're reaching out to raccoons because in their natural environment, the raccoon does have a brain that's closer to a primate brain. It looks like a primate brain. They're not primates. All this gyrification, and they use their forepaws like we use our hands. And just one quick little finding, that the raccoons that we're looking at, we're working with the USDA in Colorado, they have captive raccoons doing rabies research, and animals that perform better on problem-solving tasks have more cells in their hippocampus than the non-solvers. So we're looking at individual differences using that technique that Dr. Herculana Huzel uses to count the cells in the brain. So we're working on writing grants to learn more about the raccoons. So that completes our brain and experience part of our journey. And I will throw it to Jim to see if he has any questions that I can answer before we go to the rat driving.
Okay, thanks, Dr. Lambert. So do rats continuously generate new cells, and do they need to continue the process of having additional stimulation to keep generating new cells?
Yeah, that's a great question. We do think that rats do generate these cells, and they're not only in that hippocampus, but around the nasal area, the olfactory area of the brain. We need to know more about it, but we have found that if you put old rats into enriched environments, kind of couch potato rats, and they start to exercise and to engage in these with the stimuli, that they can increase their cells, and not only increase cells, but increase the connections. There was a human, it's a bit more controversial with humans. We know that we have more neurogenesis, the production of new cells, earlier in life, but it also looks like they continue to be produced throughout our lives, but the building of new connections is just as important. So neuroplasticity, I like to say, goes from the womb to the tomb. We are all accountable. That's what I tell my students. We're all accountable throughout our entire life of what we're doing with our brain to keep it healthy.
Okay. Which mammal has the largest brain, and which one has the most neurons?
So when you get into dolphins and whales and elephants, they have much larger brains than we do, and they have more neurons. But I'll use as an example, the elephant, more of its neurons is in that back, well, more of our neurons are in the cerebellum hanging off the back of our brains, but elephants have a lot more in that area than we do. We seem to win the contest, not in total number of cells, but in that cortex. So if you ask which animals have the denser population of cells in the cortex, humans are going to win. But those large mammals, large animals, like the elephant, have a much larger brain and more neurons. So the distribution of neurons is important, what area of the brain it's in. So that's a very wonderful question that I could spend a lot of time. Dr. Herculana Huzel's book is wonderful for looking at the comparative analysis if someone's interested in that.
So if I could just do a real quick add-on to that for myself, where do like corvids, like really birds like ravens and jays, how do they fit into something like that?
So the bird brains are somewhat different. So they certainly have great, and parrots too, very impressive abilities. But the distribution, the brains are obviously smaller. I'd have to look to see how their cortical distribution, the human still wins that contest, but several of the bird brains are quite impressive. but it's a little bit different distribution. But their problem solving is amazing. So it's a great question.
Okay, and one last one. How did you become interested in rats? Did you have any interest in rats before you started studying them, or were they just a good kind of model for your research?
Well, as a kid, I was that kid out in the woods picking up mice, I guess mice more than rats, just interested in any type of nature and animal. So I'm a huge animal lover and interested in that. As a neuroscientist, I was trained with rodents, so that was the journey that I took. But I love the rodents. I have a lot of respect for them. They're incredibly smart, even though they have a tiny brain and not so many. The neurocircuitry is not as impressive and as sophisticated as the humans. But I wrote a book, the Lab Rat Chronicles, about how smart these animals are. So it was maybe an arranged marriage, but certainly was a productive one. I continue to be fascinated with rats, although I know to learn more about the brain, I need to get out and look at other animals and look in more natural environments. And that's what we're doing with the raccoons. So
Okay.
should we go on?
Yes, thanks, Dr. Lambert.
All right. So we want to get to a different part of our research road trip and think about driving. So a little bit of history, because this is not a natural behavior. You just heard me talk about naturalistic behaviors, and this is not. I've never seen a rat out in the alleys or the woods driving cars. But science is wonderful and collaborative, and I have to thank Dr. Beth Crawford. When I came to the University of Richmond, Beth approached me. She's a cognitive scientist, and she had seen a museum exhibit in museums where goldfish would swim in a certain area of their aquarium, and by swimming to that area, they could move the aquarium. So they're kind of driving the aquarium. And so she's very creative and thought, I wonder if you could teach a rat to do something like that. So she asked me, Kelly, do you think you could teach a rat to drive a car? And I thought, no, why would I want to do that? That's not naturalistic. But she was very persuasive, and we started thinking about this, and she's interested in this. We started talking about very deep terms such as embodied cognition, where cognition is not just a product of the brain itself, but it emerges from the brain's interactions with the environment. And could we expose the rat to a more sophisticated task, and would that change their cognition? Would we learn more? And if we're interested in translation, humans drive, so why not? I was in. So I was packing up to go on this trip. Before I knew it, we were talking about how to design a car. I brought to the table somewhat expertise with rats after working with them for 3 decades. She brought to the table, her cognitive science and programming skills. So we thought at the beginning, I thought that the rat would use its nose to activate the bars to drive the car, and it's standing on a plate so that when it touches those bars, it would complete a circuit and drive the car forward. No, their noses are very sensitive. I shouldn't have thought that to begin with, but they can grab those bars. And that's what they did to drive the car. We also needed something for them to work for, and they love Fruit Loops, so we habituated them to Fruit Loops. And before I knew it, and it just gets me every time, they were getting in that car on their own after knowing they could get a Fruit Loop and driving to what we call the Fruit Loop tree to get that reward. It took a lot of patience and training and something called operant conditioning. If you have studied psychology, one of the fathers of American psychology, B.F. Skinner, introduced this term in his learning research based on this word operant from the word operate. So looking at how an animal operates on its environment, that determines and influences behavioral patterns. So if a response leads to something rewarding, an animal is more likely to do that. So we use operant shaping and operant conditioning to get the animal into the car, and then to grab those bars to get to the Fruit Loop, eventually the Fruit Loop tree. So to bring in the first part of our journey with this second part of the journey, I was very interested if the animal's experiences up to the point of where we started training the animals would influence their driving skills. So we took animals that had been raised in an enriched environment, and those that had been raised in a standard environment. And with those forward driving, that car that I just showed you, we simply tracked how many trials or days of training it took for it to drive all the way to the Fruit Loop tree. Get in, drive till the end. And it has to hang on. It can't just touch the bar. It has to hang on to get there. When we compared that to the standard animals, we just gave up. In this experiment, they never learned. So that said a lot. Their experiences going up to this task determined whether or not they could learn the task. So the enriched animals, yes, they got their driver's license, but in this case, the standard animals, they were denied. They did not. So our experiences leave this imprint on our brain. We were able, this most creative project I've ever tried to write up, but we were able to publish this. And Dr. Crawford's team decided to take it a step further and teach the animals to steer those cars, not only drive straight. So you see that they've added bars on the side. So they turn to turn the car in that direction, and then they have to reposition, restart the car anywhere. So they're not just memorizing a task, they're having to do it differently. So this is a very different cognitive ability than I have ever seen in the lab. They're using their, I'll call it a skill, to maneuver and navigate that car to the reward. So we were all geared up to follow up on this research and some other research we had in the lab planned for this summer. Typically have 6 or 7 undergraduate students working in my lab. We were working really hard, and then, something happened. We hit a roadblock, and that was the COVID-19 crisis. So we had to recalculate our plan. I could no longer have animals in the lab, but not students. So I do have a team where we were trying to think of what we could do. We needed, through this recalculation, we expanded our crew. I reached out to a former colleague at Randolph-Macon in the computer science department, Dr. John McManus, who does wonderful work with programming and robotics. He had a student who was interested in this project, Max Spivey. See him there with his spider robots. Maybe that should be at University of Richmond, the spiders. So we're trying to figure out if we could do something remotely, designing cars, and Bilal Hindi is a recent graduate from University of Richmond, so I was able to hire him as a postbac so he could come into the lab. And then working remotely is a current student, Sarah Hugh. She has interest, she's a psychology major, but with interest in also triple majoring in computer science and physics. So she was very interested, and she was on the initial team training the rats to steer. So in our recalculating, we got together via Zoom. I'm going to show a little video of that process to think about what our next step was. Our next step was, we were interested in this steering and navigating and this embodiment. And in our initial studies, the animal had to actually turn in the direction it wanted to navigate the car. Now we wanted to rebuild a dashboard and kind of redesign our car. It's a little bit more streamlined now, bringing the computer scientists on board. And we have levers all in a dashboard situation. So they press the middle lever to go forward, and the right to go right, and the left to go left. Which going to be a big leap. We're still in the process of doing this. So they're learning that I'm going to turn this car without turning my body. That's so interesting. So we've printed and built different versions of lever pressing and dashboards. We were also interested in whether or not the animal could learn to press down or forward, if that made a difference. We had a forward driving car or forward lever and a downward lever. So lots of interesting questions as we went in. We also added a little camera inside so that we could do a micro analysis or look at micro behaviors as the animal gets in there and it's eating Fruit Loops. We're still in the shaping stage, but it can press. In this case, we train on a middle lever to go forward, and then we're going to add the right and the left. This takes a lot of patience, but having this camera really is wonderful to see what they're doing up close and personal. So with every journey road trip, it's good to stop along the way and have a little fun. So I'm going to tell you a little bit about the data that we found this summer, but one of our research technician, Olivia, put together a little video to show our discussions, the printing of the car, and some of the training with a little driving music. So I hope you enjoy this. So that was our little fun journey, and that gives you a glimpse of some of the challenges we had trying to do a research project this summer, but it makes you more creative to think of things differently. But we really appreciate the collaboration with Randolph Macon in designing those cars, Dr. McManus and Max and the team here to help train these animals. So what have we found out with our new dashboards so far this summer? I told you that we were looking at different types of dashboards, and we found out that the downward pushing lever is much more, the animals are much more predisposed, and they train more easily with that downward pushing lever. I should have also mentioned that we're continuing to look at the type of environment. So we have animals, and our ends or numbers are very small because this takes a lot of training to do. We have animals from the loft, the enriched, and the standard environment. This is days until driving. In this case, you see the lofts are driving much faster than the enriched. And the standards aren't, again, they're not picking this up as readily. So the complex environment gives the animals a little bit of an edge. It doesn't mean they can't learn it. Looking at the number of days until the animal would get the Fruit Loop in the car. I was interested in this because Bilal, who was training, said, Dr. Lambert, the standards are not eating the Fruit Loops in the car. And you see that there's a big difference between the complex environment and the standards. This suggests that the standard animals are not comfortable enough in the car to eat that reward. So that means that, as I mentioned before, these complex environments are probably also influencing the stress response. And that can influence learning as well. In fact, our standards have started to drive without eating a Fruit Loop. And I'm fascinated by this because you usually are thinking about, okay, what is the reward? What is the incentive? So we think that the enriched and locked animals, they're driving because it's very clear they want that Fruit Loop. You saw that straw coming into the video. They're driving to the Fruit Loop tree to eat that Fruit Loop. But if the standards are driving some now without the Fruit Loop, that suggests that they may be trying to escape from us. I told the trainer, no offense, but they may be trying to get away from you. So this could be a difference between incentive-based, positive kind of learning versus fear-based learning. And that gets us into a territory that I've never been into, looking at individual differences in that way. We could expose them to different things in a different way, like threats or something, but they hadn't been exposed to threats. So that's been fascinating. And then looking at some of the behaviors in the micro with the dashboard camera, Sarah has, so far, it looks like it may be an interesting way to look at what we call lateralization. So like the different sides of your brain and different, like right-handed or right-pawedness and left. She has noticed that the animal, when it's using its right paw, will kind of point the nose in the direction of the left, like you see here, more of a balanced lateralization. But those are the questions that we're looking at. And so the downward pushing, we've converted to only downward pushing cars. We still haven't got to the question where they're steering to see if they need to turn their body to steer when we use a template similar to the first car versus our forward dashboard. So these are questions I never thought that I'd be asking, but it really, to train the animal to do a complex task, allows us to ask very interesting questions. And in fact, one researcher who was working with Alzheimer's, models of Alzheimer's, suggested that we needed to use more complex tasks like this to be able to enhance the translation from the rodent work to the human work. So it's been really exciting. A different type of summer with a different journey than we thought, but very exciting. We're getting through, the best we can. We also did something different. We had a summer research book club, and we read, our driving team, Dr. Matt Crawford's Why We Drive Toward a Philosophy of the Open Road. I highly recommend this if you're interested in driving, and if you're interested in the brain and the mind and philosophy. Dr. Crawford is a philosopher who loves cars and motorcycles, so it's an interesting mix. If you recognize this name Crawford, you may think, I remember Beth Crawford. Yes, that's Beth Crawford's husband. And one of the chapters that we loved, it was my favorite chapter, is entitled Project Rat Rod, and that's a chapter on the first version of the driving rats. And at the last paragraph, as we're thinking about what we can learn from the rats, learn from driving, he discusses and is considering this question of driverless cars. He loves the control and the feel of the road, and it's an interesting question, this cost-benefit analysis of a sense of control versus safety and efficiency, and looking at what we've learned from the rats. So I wanted to read this last little bit here as we're closing. Flourishing, and that's what we all want to do, right? To flourish. That of rats and humans alike seems to require an environment with open problem spaces that elicit the kinds of bodily and mental engagement bequeathed to us by evolution and cultural development. These exquisitely honed human capacities include the glorious century-long development of the automobile. He's a fan. That astonishing tool, and the social intelligence that we have brought to bear on the problem of sharing the road together. If instead we put ourselves in a plexiglass enclosure, like the standard rats, in which all our most basic needs are met, we will have nobody but ourselves to blame if we begin to feel like standard lab rats in a massive laboratory of social engineering. We would be safer that way, no doubt. But this is my favorite line in the book. But remember, or 2 lines, but remember, all rats die. Not every rat lives. So even through this pandemic, we're learning that it's important to live, to engage, to live in this complex world. And in the acknowledgments, it was really cute that he acknowledged the first 2 driving rats, Mario and Luigi, and thanked them for persevering to get to that Fruit Loop even when it was quite difficult. So this may be good to add to your end of the summer reading. And if I could take a minute, as this is my last slide, to incorporate this into what we're experiencing now. I mentioned that life changes and circumstances change. And when we're training our rats, we have a habituation phase where we slowly get them to go into that car and say that it's good, you get a Fruit Loop. Sometimes the world changes and we don't have that habituation period. All of a sudden, we're in lockdown, we're in quarantine, everything has changed, we're wearing masks and we haven't done that before. So in an attempt, kind of a public service announcement, a way to incorporate what we know about the fundamentals of behavioral science, our lab, I guess I, along with Katie McBride here, she's on our design team at the University of Richmond, have brought this character, a raccoon, you already know that I like raccoons, to life. We're calling this character Kalo. Kalo is from the Greek word meaning help or to make whole. Raccoons already wear a mask and they like to wash their forepaws. Actually, they're foraging in the water, but they have their forepaws in water. So they're a good model. And they're using science as their superpower, and that's perfectly appropriate for a science museum talk, to try to encourage children to follow Kalo's lead as the natural behaviors to win this battle against COVID-19, to wear this mask, to wash their hands, and to distance. We have a website, kalothehero.com. If you know teachers or have kids who are interested, we have downloadable materials, and this is just, we're not supported by anything now, it's just a passion that we have. So kids can download a pledge to take Kalo's pledge to engage in these behaviors. There's behavioral monitoring sheets where they can monitor each day they engage in these behaviors. They can become their own behavioral scientists. So we're incorporating incentives and positive associations and a sense of agency. And we want to incorporate a little bit more about what to look forward to as we get past this COVID-19 crisis. It's important for that dopamine to look for a time when this is over. So we're trying to incorporate what we've learned from the rats and the environment and behavior into something that may help kids and their parents and all of us adapt at this time when our environment has changed. So we've taken a lead from the Smokey Bear campaign in a way to do this. But we have a Facebook page in this, and I'm happy if you email me, klambert@richmond.edu. Happy to provide these materials or answer any of your questions. So I will throw it back to Jim.
Okay, Dr. Lambert, we have got quite a stack of questions lined up here, so we'll get to as many as we can as many as we have time for. So we've had a couple variations on this question. Here at the museum for our rat basketball team, we use just strictly female rats. So anyone who's caught rat basketball here at the museum, you've seen all of our rats are female. So is that true of your research also, or have you used male and female rats?
Good question. We could use either. The first, Mario and Luigi, were males, and they did fine. The subsequent animals that we have used have been females, but for a very practical reason, they're a little bit smaller, and the big older males can't fit into the cars very readily. So their learning ability, and you can talk about subtle differences between males and females and their brains and behaviors, but both train well. I guess I'm not sure if I knew that you only use females for the basketball. That's what you said?
Yes, ma'am.
So certainly they're doing, I should have mentioned that the rat basketball is a huge hit, or used to be. It's a huge hit at the Science Museum. And again, they're using all these fundamentals that I just talked about to shape, to train, to reward those animals. So if you want to train an animal, male, female, that's level playing ground, but if size is important, if you have an animal where the males are larger and you have sexual dimorphism, then you may want to go with the females. Sometimes the males, if you're housing them together, can get a little bit more aggressive, so that could be an issue too.
Okay, let's get to another one here. So the treat tree, where the rat was driving, it seemed like the rat had to just hit the wall in order to stop. So have you thought about maybe putting a treat tree in the middle and seeing if the rats could learn to stop on their own to get to it?
Yeah, that's a great idea, so that they don't hit it. I don't think they have whiplash or anything. It's a very soft border barrier. But yeah, we could maybe extend something from overhead so that they wouldn't have to go to the very end. We've talked about doing a backup and maybe even brakes too in the future. But it's a little bit of a rough ride, you're right. It's like me parallel parking. So thank you for that suggestion. I'm going to write that down.
Okay. So actually, again, had a couple people with this one. So a couple people with rats and a 3D printer and people who know how to program. Is this something that they could basically do at home and teach their pet rats how to drive?
I have, maybe I should have mentioned when we published that, we got a lot of media attention. So I think our PR group, there was 1,500 stories were published about these rats. So I had a lot of emails of parents, of kids, of teachers, science teams. So we did, Dr. Crawford published instructions and something called, a website called Instructables. So you may want to look for that. However, we've changed with now 3D printing the cars. We've stepped that up, but we will be working on getting some instructions when we write this up. So I can connect you to Dr. McManus if you want to email me or email him to give some general instructions there. That's more in his wheelhouse than mine. But you could get started by looking at the Instructables and rat car, and you can contact me if you can't find that. So it's a good project for kids and such. And it's not just, I had one person in Germany who built the car, but he said he was using degus, an interesting rodent. They're not driving. And say, oh, well, you still have to teach them how to drive. They don't you don't just give them the keys and they automatically drive. So it does take a lot of patience, but it's a good learning experience on all levels.
Okay, so we had another question with the rats that were driving without the Fruit Loop that maybe they're trying to get away. Could it possibly they just like driving? Is it possible?
That's our second Plan B hypothesis. So that's very insightful. The two solutions. One, I mentioned the fear because they're not eating the Fruit Loop there, but they could also enjoy the movement of the car. And that was the number one question we got with the first batch, our first research. Do rats like driving? And that's a great question, but it's hard to answer. So we have some variations about, we did let them put the car in there in our first research, and we didn't reinforce them. And so they would drive for a while, and they're enriched more than the standard rats. So they will drive, but it seemed they lost interest after a while. So right now it's looking like it's not an automatically or it's not very naturally rewarding, but we have not looked as thoroughly into that question. So if the animal's driving, it has to be for a reason for those animals, not for a Fruit Loop. So it's either for an escape or the movement itself is rewarding. So that's a very insightful observation.
Okay, so I think we've maybe got time for one, maybe two more. So you mentioned your book, The Lab Rat Chronicles. What's the reading level for that? Is it appropriate for like young adults or?
Definitely for young adults and adults. It's written for adults, nonfiction, researchy kind of, but it's very, I'm in education, so I try to make it very readable. And so small kids, if they're, depending what their reading level is, but certainly from junior high up would probably, if they like animals, I think could certainly follow it.
Okay, so last one. So you mentioned using a variety of different tools and different stimulations for rats. For translating the findings to human cognition, how do you kind of do that?
Yeah, so it's interesting to think about what if we're living in an enriched world, especially as we've been in quarantine and our world has become much more digital and focused, we're doing experiments on ourselves, so I don't have the answers to this, but are there certain elements of our environment? Maybe digital, being in front of a computer is okay, but do we still need the physical touch and the social interactions? Research does suggest that we do. So I think at one level, we're showing that yes, with this simpler model of a brain, changing the environment changes the structure of the brain and changes the potential output, the perception of affordances. So we've shown that in a laboratory model. So that tells us that, gosh, if we have people in prison, that's a standard laboratory environment. Are we taking individuals who need adaptation for their brain and stimulation and rehabilitation and doing the worst possible thing for their brains? Lower socioeconomic status when we don't have a lot of engagement, perhaps, I mean, sometimes, or being in quarantine. One of my colleagues were writing a grant together in Neuroscience. He said, I just feel stupid in quarantine because I'm not engaging my brain as much. And it looks like we probably need to do more than the digital engagement. So we're taking a model, learning just more about that fingerprint, that imprint that the environment places on a brain. And knowing that, I think that gives us more recognition, and we need to be more respectful of behavioraceuticals, as I call it, to make it seem more medicinal. But that could be, I think, just as impactful as drugs and surgery and other more traditional medicinal options. So we have to be careful in translating, but it's important to have this perspective, I think.
Okay. Dr. Lambert, thank you so much for being here. It looks like we're out of time. Thank you all for joining us, and thanks to all of our speakers for helping us discover more about our world. Please join us next week, August 26th at noon, for Pesticides in Perspective, presented by Dr. Wayne Searles. He is a strategic consultant in agricultural, apiculture, green industry, and structural pest control markets. So you can register for next week's talk at smv.org. Each talk is free to attend and is open for the first 300 registrants. Thank you for joining us today. Thank you for our sponsors at Bon Secours. Until next week, stay safe and stay curious.